Yeah, thanks, Anke. Thanks, Lucas. And as Anke mentioned, the next speaker is Paula Rio. I'd like to welcome her to the stage. She is currently the head of the bone marrow aplasia unit at the CIEMAT Research Center in Madrid, in Spain. She's worked there in various roles since 2005. Originally, Paula studied biology at the Complutense unit in Madrid and got a PhD in 2002. Since 2010, she's been working in gene editing, focusing on gene therapy approaches for Fanconi anemia, which is a rare genetic disease caused by defects in DNA repair pathways. Paula has both pursued basic research in how various gene editing technologies can be applied for that particular condition, but is also involved in 2 clinical trials for experimental treatments. So please welcome Paula, and we're excited to hear her talk.
So good morning, everyone. Thank you very much for the invitation to present our results here. So today, as Florian has mentioned, I will talk to you about the gene therapy approaches that we are following to correct Fanconi anemia hematopoietic stem cells. So Fanconi anemia, just as a brief introduction, is an inherited bone marrow failure syndrome. In general, it's associated to autosomal recessive mutations in 22 different genes that are called FANK up to FANK-W. The most frequently mutated gene in the Fanconi anemia patients is FANK-A, and all the genes cooperate to generate proteins that are participating in a pathway to repair a very specific type of DNA damage that generates interstrand cross-links in the DNA. At the clinical level, the patients are usually show congenital abnormalities, an increased cancer predisposition, especially to squamous cell carcinoma and also leukemia. But the main and the first symptom in the patients is the bone marrow failure. So the hematopoietic stem cells start to decrease. They cannot proliferate and generate the other cells in the peripheral blood. And finally, the patient develops bone marrow failure. And the only curative treatment for this bone marrow failure is the allogenic hematopoietic stem cell transplantation. But as probably most of you know, it's challenging to find a suitable donor for these patients, and also the conditioning and the graft versus host disease that is generated in these patients leads in the future to an increased incidence of squamous cell carcinomas in these patients. So for that reason, in our case and other groups have been for many years trying to develop gene therapy strategies to correct the hematopoietic stem cells from the patients and reinfuse them in the patients. Initially using lentiviral vectors, and nowadays in preclinical studies using gene editing approaches. I want to show you the results of the gene therapy trial using lentiviral vectors, because I think it's a way to understand why we want also to move to gene editing approaches in this disease. So in 2016, we started a phase 1-2 clinical trial, gene therapy clinical trial for Fanconi anemia A patients, that, as I mentioned, is the most frequently mutated gene worldwide, but also in Spain, where we have around 80% of patients carrying mutations in this gene. And the idea was to obtain hematopoietic stem cells from the patients early in the diagnosis, after the diagnosis of the disease, because, as I said, a main limitation in this disease is to obtain enough hematopoietic stem cells from the patients. We could select the hematopoietic stem and progenitor cells population by the immunoselection of the CD34 positive cells, and these cells were transduced in a very short period of time using, in this case, a lentiviral vector carrying the wild-type FANK with an exogenous promoter. And these cells were infused in the patients in the absence of conditioning. As probably you know, for other clinical trials, for gene therapy in hematopoietic stem cell disorders, usually you need to do conditioning because you need to make a space in the bone marrow of the patients to engraft the new cells, although they are coming from the patient. In our case, we thought conditioning would not be necessary for the patients because they have a low content of hematopoietic stem cells, and the corrected cells should have a proliferative advantage and in comparison with the uncorrected ones. So we treated 9 patients. In one of the patients, the drug product didn't meet the release criteria, so we didn't include in the analysis. And as you can see here, this is the analysis of the peripheral blood in the different patients over time. You cannot see, sorry. Over time. And as you can see, we infuse, of course, different number of cells depending on the number of cells that we could harvest from the patients. And what we have observed is that, in general, the engraftment is associated with the number of cells that we could infuse. If we don't consider this patient in which, although we infuse a high number of corrected cells, we can hardly detect engraftment, and this is still under analysis because we don't understand the behavior in this patient. But in the rest of the patients, as you can see, for example, in patient 2 and patient 6, we observe initially a very low percentage of corrected cells, since most of the cells are the endogenous ones. And when we analyze over time, we started to see the increase in the corrected cells over time, reaching, in the case of this patient, almost 80% of the peripheral blood cells corrected by gene therapy using the lentiviral vectors. When we perform the functional studies, so we analyze the correction at the level of the bone marrow, hematopoietic stem and progenitor cells by the use of an interstrand cross-linking agent, that is the mitomycin C. The cells from the Fanconi anemia patients, as you can see before the patient entering the trial, are very sensitive to this interstrand cross-linking agent, and they recover the resistance after the engraftment of the corrected cells, reaching again, in the case of this patient, levels very similar to the healthy donors. We also analyze the reversion of the phenotype in the peripheral blood cells from these patients, and this is the test that is usually used to diagnose the Fanconi anemia patients. So the T cells are pre-stimulated in vitro and also damaged with an interstrand cross-linking agent, the deoxybutane, and the percentage of aberrant cells is analyzed. So you can see again that all the patients have a high percentage of aberrant cells when they enter in the trial, and this is decreasing over time, reaching, in the case of patient 2 and also in patient 4, levels similar to healthy donor. This means that if this patient has to be diagnosed nowadays for Fanconi, he could not be diagnosed as a Fanconi anemia patient anymore. So what about the improvement in the hematological counts in these patients? So what we have observed is that in those patients in which we have engraftment of the corrected cells, but the engraftment has been delayed over time because of the number of cells that we infused in these patients was a bit low, we could not observe the reversion or the stabilization of the bone marrow failure, and the patients require other treatments. However, in those patients, in patient 2 and patient 6, in which we infused a higher number of cells and we engrafted the corrected cells early enough after the development of the bone marrow failure, we have observed an improvement in the hematological parameters in both patients, or an stabilization in other parameters. As you can see in the dot lines, we show here the natural history evolution of the Fanconi anemia patient. So this would be the expected evolution of the disease in this patient with the age of the patient, and we have improved the natural history by the use of gene therapy with lentiviral vectors. So the main conclusions from this gene therapy trial is that so far the lentiviral vectors seem to be safe in this case, although, as you know, the integration is semi-random with these vectors. And the good point is that for the first time we have seen that corrected CD34 positive cells from Fanconi anemia patients can be engrafted, and they correct the phenotype of the hematopoietic stem cells in the patients. And if we do this with enough hematopoietic stem cells and early enough during the development of the bone marrow failure, we could correct the disease in the bone marrow failure in these patients. So with this idea, of course, when gene editing came in 2012, we started to think about the possibility to use gene editing to correct these cells, because in comparison with the lentiviral vectors, at least in theory, we could have a control correction of the gene, and also this will allow us to maintain the endogenous regulation of the gene, something that it doesn't seem to be relevant, for example, in the case of Fanconi, but in other Fanconi anemia complementation groups could be also relevant. So, as most of you know, and also Florian has mentioned, when we think about using CRISPR-Cas technology to correct disorder, we usually think about the possibility to use homology-directed repair. Jacob Korn's group and also our results in the lab show that this is not really efficient in Fanconi anemia cells, that homology-directed repair is partially hampered. And also if we think that our aim is to correct hematopoietic stem cells are quiescent cells, so the efficiency of homology-directed repair is markedly decreased in comparison with non-homologous enjoining. So our idea was, could we use the non-homologous enjoining to correct specific mutations in the patients, although in general this strategy is used to generate knockouts. So we wanted to mimic what is happening in some of the Fanconi anemia patients, in which a phenomenon called somatic mosaicism is occurring. So if, for example, this is a patient, it was published in 1999, in which the patient has a deletion of one nucleotide, and due to the high chromosomal instability, a second or even a third mutation can occur close to the initial mutation, and this can lead to the restoration of the open reading frame. If this happens in a hematopoietic stem cell, this leads to the recovery of the hematological parameters in this patient, as happens in this Fanconi anemia patient from our cohort of patients. So with this idea in mind, our hypothesis was, can we use the CRISPR-Cas technology to perform similar, to generate a somatic mosaicism in cells from patients? So these are the mutations that we can find in the Fanconi anemia A gene in the patients from Spain that was published by Jordi Surrayes' group. And we selected the most frequent mutation in the Fanconi anemia patients from Spain. 30% of the patients carry this mutation. This is a premature stop codon in exon 4. This was a challenging mutation to correct by this approach because we don't only need to recover the open reading frame, as is the case in most of the mosaic patients, we also need to remove the premature stop codon. And we have to do this without disrupting any functional domain, something that was not very clear at that moment because the protein was not really well characterized. And there is no description of any reversion in patients carrying this mutation. So this could indicate that this region of the gene was really essential for the function of the protein. But anyway, since we want to develop a therapy for the patients, we wanted to test this approach. So we designed 2 different guide RNAs to target the region close to the mutation. This is the mutation, and these are both guides. We use them separately and tested initially in lymphoblastic cell lines and afterwards in hematopoietic stem and progenitor cells from the patients. I show you here the results from obtained in CD34 positive cells from the patients, in which in 3 different samples, when we perform the editing and analyze the indels generated, we observe that between 0.4 and 8% of the editing events were potentially therapeutic indels. This means that eliminate the stop codon mutation and restore the open reading frame. When we maintain these cells in culture and generate hematopoietic progenitors, what we observed was a marked proliferative advantage of these potentially therapeutic indels in all cases. And when we analyze these editing events, we observed that a deletion of 30 base pairs was restoring the open reading frame and leading to the proliferation advantage of the corrected cells. And this deletion, of course, eliminated the stop codon and recovered the open reading frame of the gene, suggesting, and something that was confirmed later on, that was this new protein can correct the function of the Fanconi anemia hematopoietic stem and progenitor cells. But of course, if we want to use this strategy for clinical application, the initial efficiency was really low. So we designed a new approach in which we can use 2 different guide RNAs, one of them now closer to the mutation, and generate a specific deletion of 18 base pairs. When we use both guide RNAs together, we can obtain an initial editing efficiency close to 30%, and again, when these cells were maintaining culture, this is increasing a lot. So 28% is much more closer to the percentage of corrected cells that we could obtain in the clinical trial using lentiviral vectors. So good fit for the application of this approach. So what is the other advantage of using non-homologous enjoining gene editing in comparison of lentiviral vectors? Well, also that we can use for other mutations. Sorry, this is only to show you really that in the cells corrected with in the cells edited with both guide RNAs and that show a proliferative advantage, we have also observed the re-expression of the Fanconi protein and also the reversion of the sensitivity of these cells to mitomycin C, what confirms really that they are functional. So as I was mentioning, so the idea or the advantage of the non-homologous enjoining is also that we could use this approach to target other genes. So if you think about generating a lentiviral vector for each of the 22 different Fanconi anemia genes for groups that, for example, only represent less than 1% of the patients, this is really cost ineffective. So, and also in some of the cases, lentiviral vector production is very complicated for genes like BRCA2, that is 10 kb. So the production is really challenging. So we show that for specific mutations, of course, we can target by non-homologous enjoining other Fanconi anemia genes as FANB, FANC, FAND2, and BRCA2, and with initial efficiencies, since in this case the mutations were only point mutations in the patients, initial efficiencies that can be applicable in therapy. In the case of BRCA2, this is also very relevant because BRCA2 patients cannot be corrected by homology directed repair, but because it's not only reduced, it's completely hampered in these patients. So at the moment that we use this approach, this was the only way really to correct BRCA2 hematopoietic cells from the patients. What can be the other advantage of the non-homologous enjoining? As I initially showed you, the possibility to efficiently correct hematopoietic stem cells in comparison with homology directed repair. And that's what we saw when we analyzed the editing efficiency using CD34 positive cells. When we edited these cells and analyzed the different subpopulations of hematopoietic stem cells, we observed that the editing was very similar in the total CD34 positive cells and also in the most primitive hematopoietic stem cells that are really our target for any of the therapies that we are developing so far in Fanconi. And when we transplanted these cells in immunodeficient mice, we observed the same, that a long-term post-transplant in secondary recipients, the edited cells were maintained over time, indicating that we can correct this rare population of hematopoietic stem cells that will last in the patients for all their lives. But of course, not all the mutations can be corrected by this approach. We estimated that around 40% of the mutations could be corrected if we are not hampering or affecting any essential domain of the gene. So in collaboration with Jacob Skorm lab, we started several years ago a collaboration to test the possibility to use double-strand break-free methods to correct hematopoietic stem and progenitor cells from the Fanconi anemia patients. Also because this is really attractive in the case of Fanconi because we are talking about a DNA repair disease. So initially, we focus on base editing. Jacob will explain you in more detail the approach that we follow. But in this case, the base editing strategy, as most of you know, we have the possibility to use cytosine base editors or adenine base editors. We focus on the adenine base editors just because of the type of mutation that we selected that allowed us using a deaminase together with a guide RNA to modify point mutations, to modify the adenine to guanine, and in this way have the possibility to correct the cells from the patients. So just as I mentioned, since our aim is to correct hematopoietic stem cells, we in the lab optimize the possibility to target the long-term repopulating hematopoietic stem cells using the base editing approaches. So for this, we selected a guide RNA that is targeting the ABS1 locus. This is a safe harbor locus, so we are not going to hamper the engraftment capacity of the cells, just to see that we can efficiently target hematopoietic stem cells with this approach. So we combined we tested the two different base editors, ABMAX and AB8E, and as you can see, when we use the AB8E, we obtain levels of editing in vitro around 80% or even higher. So very similar to the ones that we obtained with the CRISPR-Cas system initially. Most important, when we transplant these cells, we observe that the engraftment capacity of these cells in primary and also in secondary recipients was very similar to the untreated cells. So base editing is not affecting the engraftment capacity of these cells, and is not affecting either the multilinear repopulation capacity of these cells. And even more important, what we saw is that the editing efficiency in the case of the AB8E was maintained when we transplanted these cells in primary and even in secondary recipients. So this seems, at least in my opinion, the base editing is a really amazing strategy to target hematopoietic stem cells, long-term repopulating hematopoietic stem cells. And Jacob will explain you all the experiments that we have performed in Fanconi anemia hematopoietic stem cells after confirming that this approach can be an efficient strategy to move in the future to the patients. So, but anyway, still we need to correct other mutations from the patients. So we also focus on the possibility, when this strategy was published in 2019, to use the prime editing to correct hematopoietic stem and progenitor cells from the patients. This is really, when I read about this strategy, I thought this is really amazing. And that's the case, but of course it's also, or it has been also challenging. So what are the main advantages of prime editing? As I mentioned, in the case of base editing, that it will not generate double strand break in general. We don't need the donor template. This is also an advantage in Fanconi because the delivery of the donor template is also challenging in Fanconi because of the toxicity associated to the delivery. We can correct the sequence to wild type, and we can generate a lot of different modifications, small insertions, deletions, and point mutations that cannot be corrected either by base editing or also by non-homologous enjoining strategies. So in this case, the main advantage of prime editing is that you combine the use of any case with a reverse transcriptase, and you can use a guide RNA that carries both the sequence that will be recognized in the DNA sequence where we can generate the edit, and also the region that we want to edit. So we can include in the guide RNA this sequence, and this will allow us to retrotranscribe this guide RNA and integrate into the genome and substitute the mutated one. If we also use a guide RNA that is nicking the other strand, we can increase the efficiency of this approach using the so-called PE3 system. So we tried to establish these approaches initially in healthy donor cells, in hematopoietic cells, K562 cells, in which we optimize this strategy using a guide RNA that is not generate a point silent mutation. And also combined with the use of a prime editor, an optimized version that is the PEMAX. In this case, we use the conventional system, the PE2 and also the PE3 that, as I mentioned, use a nick, a guide RNA that will generate a nick in the other strand. When we tested this in healthy donor cells, what we observed is that the PE3 system leads to higher efficiency, around 40%. Also this when we combine with the PEMAX, that is really changing the efficiency of prime editing in comparison with the initial prime editors described. Also, in 2021, it was published that the mismatch repair pathway could suppress the efficiency of prime editing so that you can use a dominant negative of MLH1 to increase the efficiency of prime editing. So we tested this initially in healthy donor cells. We haven't observed an improvement in the editing efficiency by the downregulation of mismatch repair, but I think that also this can be associated to the toxicity of using so many mRNAs to deliver in the cells, and we are trying also to work on this. But with the initial results that we obtained with the prime editing 3 system, we thought it would be enough to move to the Fanconi anemia cells, and also Jacob's team designed a guide RNA to target as a proof of concept the same mutation that we show for the non-homologous enjoining and also for the base editing, in which we can correct the stop codon to a wild-type sequence. Using again the P3 and the P5 system, what we have seen is that we can edit around correct to the wild-type sequence around 40% of the cells. And importantly, again, when we maintain these cells in culture, they show a marked proliferative advantage, suggesting that they are really truly corrected. But of course, we confirm this by Western blot for FANK. We restore the expression of FANK, and also we restore the monoubiquitination of FAND2. As I initially told you, these proteins cooperate in a common pathway, and FANK is essential to monoubiquitinate FAND2. And this allows FAND2 to go to the DNA repair foci where the DNA repair is occurring. As you can see in the cells from the patient, we cannot observe the FAND2 foci, and this is only observed when the cells are corrected by prime editing. And again, we observe the reversion of the sensitivity to mitomycin. So we also have analyzed the potential of targets because we are using 2 different guide RNAs, the PEG RNA and also the nicking guide. And when we explore the potential of targets by in silico analysis up to 3 mismatches in the genome, what we have observed is that there is no off-target activity in the sites that we have analyzed so far. So, as I was explaining all the time, it's not so easy to deliver these strategies in cell lines, but what is really important for us is to have an efficient approach to correct hematopoietic stem cells. So we moved to cord blood hematopoietic CD34 positive cells and test the efficiency of prime editing in these cells. So far, we have obtained around 20% of editing efficiency in this population. I think that we can improve this, so we are also working on this. And what we have also tried to see is if we are really, by prime editing, because this is something that is not described, also if we can target the hematopoietic stem cell and not only the progenitors, because as I mentioned, this is essential for a clinical application of this approach. So in this case, what we have done is to edit by prime editing the CD34 positive cells and sort the different subpopulations, the hematopoietic stem cells, the multipotent progenitors, and the committed progenitors. And what we have observed is that the editing efficiency by prime editing, this is a media of 5 different experiments, is very similar in the most primitive hematopoietic stem cells and in the more committed ones. This is really important information for the future application of the prime editing in the patients. So to finish, we think that we can develop precise medicines to correct the different mutations in the Fanconi anemia patients independent of the homology-directed repair, thanks to all these new technologies developed. The non-homologous enjoining is really efficient to correct the Fanconi anemia hematopoietic stem cells and the long-term repopulating hematopoietic stem cells. Also, this is the case of base editing, and in the case of prime editing, we have seen that it's applicable for many mutations, and we can target hematopoietic stem cells, but still we need to show this in the Fanconi anemia cells, and of course, this is the next step for us. And all the work done for many different people, the clinical trial that is led by the head of the department, Juan Antonio Buere, and Julián Sevilla, that is the PI of the clinical trial. Many collaborators from our lab, the GMP facility that we established at CIEMAT to perform these studies, and also many European, and nowadays a company that is doing the phase 2, that is sponsoring the phase 2 trial in this, with the results of our phase 1-2 trial. And in the gene editing, of course, my team. This work has been performed mainly by Laura Ugalde, that is finishing nowadays her thesis, and also supported by the head of the department. And I would like especially to thank Jacob's Korn Group, Herman Karasu, Sebastian Signer, and Alessandra Clemens, because this has been, as you have seen, a really great collaboration, and is still a great collaboration, of course. Also David Liu, because he sent us many of the plasmids and some help to perform the prime editing experiments, and all our collaborators. And I don't want to forget the most important people, that are the patients, the families, and the clinicians. We have a foundation in Spain, the Fundación Anemia de Fanconi, and they have been the ones making us to go to the clinical trial and do all this crazy stuff. So thank you very much to all of you for your attention.